I thought what we'd do today is rather than jump into the review and have to split it in half anyway, is just spend the whole day, the whole class period, looking at the lab that we're going to do on Tuesday. I think that'd be the most efficient use of our time. So we're going to do a lab called determination of molecular weight of an acid. And I've modified the title a little bit up here so that when the video is posted, it might make a little more sense. We're going to determine the molar mass of an acid. In this case, the acid is citric acid by titration with a standard base. Okay, somebody arrived. Let me see who it is. Wyatt. Okay. So I think I mentioned basic concepts of titration, but for our purposes, when at this stage in the general chemistry, when we say titration, we usually mean an acid-base titration. One of these, and it just depends on the experiment, one of these is known to be an accurate concentration. So we say we know the, let's say we know the molarity of the base, since we're titrating against an acid. We know the molarity of the base very accurately. Now, just an aside, and we'll get to it in more detail later, sodium hydroxide is not very stable over time. So once you make a standard base of sodium hydroxide, you need to use it right away. But you can standardize it with another acid, and that's what we're going to do first. Once you get this standard base, then you can measure out a certain volume, and what does that tell you? That tells you the number of moles of the base that you have in your receiving container, right? So what you want to do is, since you don't know the concentration here, right, is you want to add enough base to a known volume of acid. You can know this one very accurately. So that's the unknown. And in our case, we've got a discrepancy between the number of hydroxyls in our base and the number of protons that can be delivered by the acid. So let's see, as part of the discussion, this is the acid. It has actually 3 protons per molecule for citric acid. Let's see, 07. And these are aqueous, of course. So you'll notice that this can only, sodium hydroxide can only deliver 1 hydroxyl. The acid can deliver 3 hydrogens. So it's going to take 3 times as much sodium hydroxide on the molar scale, the number of moles, to neutralize or accommodate those 3. So we need 3 of these to accommodate 1 of those. And it produces what we expect, a salt, let's see, C6H5O7 plus 3 waters. So there's your balanced equation for the reaction. I may talk about the structure later, but right now it's not important. So here's the problem. You know the number of moles of base, but that means you only have that many moles of hydroxyls. So how many moles of hydroxyl do you need to neutralize 1 mole of that? You need 3 times that many. Okay. So we haven't introduced and learned how to use the concept of normality yet, I don't think. So we're going to use stoichiometry instead. So we can measure out a certain volume here and whatever that is, this volume times that molarity of your acid is not equal to, unfortunately, the molarity of the base times the volume of the base because of that difference between 1 to 3 ratio. Okay, so here's what you do. You know this volume exactly, and you can calculate the number of moles of base. So let's say we have, well, I don't know, just say 1 mole. Say we have 1 mole of base. How do we convert that into moles of acid? Because that's what we're going to need to calculate the molar mass, right? Moles per, mass per mole. So we got to figure out that. Right? So if we use our stoichiometry, right? Just like we always do. And then I'm going to abbreviate this as citric acid, Ca. So for every 3 of these, you can do 1 of those, like 1 there and 3 there. Right. So what we have is, once we've determined the volume of base we're using, and we multiply times its molarity, then we know that's how many moles of base that we have used to neutralize acid. So we've reached the equivalence point. All right, so now that we have this many moles of base, how many moles of acid does that represent? So this moles of base then would be here times 1/3, right? Ca over hydroxide. Right. So we actually have 1/3 of the number of moles of acid as we do base. And that's what will go in here. Okay. Now what goes in there? Well, you measure this acid is a solid, right? But you have to put it in solution to get the reaction. So you measure out very accurately on our analytical balance to 4 decimal places, whatever the method calls for, and you get a mass here, and then you titrate it, and you get moles here, you calculate the molar mass. Right? It's that simple. Every experiment that we do has one central theme. And the idea is, if you're going to calculate by the title, molar mass, you got to go back to that formula. Right? And that's how we got there. Now, that's once we know the accurate molarity of the base. That's what we have to determine first. So, well, and there's a complete discussion in here on the relationship that I've just described. The actual titration itself requires that you understand the mechanics of a titration. So let me make some room here. I'll probably have to draw those again anyway. But for a titration, what we're actually saying is, in any titration, whether it's acid-base or some other titration, we're trying to determine a value for an unknown by comparing it to values of a known. In our case, we're going to use a known concentration of base and an amount of base delivered to neutralize a known amount, a known volume of the acid, or a known moles of the acid. So in order to do that, we need a special device called a burette. It's a long tube, and it's got a valve here that you control. We call it stopcock. And then there's a delivery tip right here. down to a precisely metered size of an opening to deliver small drops. And then this tube is calibrated in milliliters. So what we find is, and that's not to scale, our particular burettes are a total of 50 milliliters, accurate to a tenth of a milliliter. So we have fine divisions in here also. Now, actually, I put that on the wrong side. It will hold 50 milliliters to this top mark, but that top mark for a burette is given as 0.0 mils. Okay, so it's inverted compared to graduated cylinders. So down here at the bottom is going to be our 50 mil mark. Now, why do they do that? It's for practical reasons. You're delivering a liquid, and you want to know how much you delivered into the receiving vessel. Okay? So if you start at 0 and you deliver until you reach equivalence, then you can read the exact volume that you delivered. That's why it's inverted. Now, if you start at 0, that's fine. If you do several repetitions of several determinations, then you're probably going to go from 0 to something here, and then you're going to go from there to there. So when you do your second one, you will take this volume, final volume, and this initial volume and subtract them. And that will be your change in volume. Okay? That's the most efficient way to do it. That way you don't have to reload the thing every time you do a titration. Okay? Now, instructions are going to tell you how to set up, how to guarantee that your burette is clean. It may look clean, but the best way to do it is after you're sure it's clean, and you'll introduce into this a certain amount. I think it's maybe 5 mils or so, 5 mils of your sodium hydroxide solution. Okay. And we're going to deliver sodium hydroxide into acid. Okay. So you just put a small amount in there, and then you can use a piece of parafilm if you want, tap it over the top. But I say, if you just put on your gloves, the gloves are clean. So I just take it out of its holder and put my palm over this and rock it. And then rotate a third, rock it again, rotate a third, rock it again. And that will coat the inside. And then you just drain it out. And then you do it again. So you do it twice. And once you've done the second one and drained it out, the second one, what you want to do is stop it when it gets right down to the stopcock. Because there's always a danger, if you let it go all the way through, what's in there? Now it's air. And once you fill it up and start trying to drain it through there, sometimes you can get a vapor lock. You can get that air bubble, and it just stays there. And the fluid just flows around it. Right? So once you've got that blown out and this is filled with your solution, then you want to stop it here so you don't have that bubble in there. Now, there are ways to get it out and I'll be around to help you if it happens. If you get a bubble stuck in there, usually it means that it's not perfectly clean. But we can't help that. I've tried cleaning them in a special solution, and I ended up eating the letters off the side of the glass. So now I just got a straight glass tube and no calibrations. So I ruined 2 or 3 of those doing that. So we have to find another way to clean them. Okay. So we've got what we call well, you know what the ring stand is, right? And on the ring stand, we're going to put this device called a burette clamp. And it fits down over the bar. It's usually shaped like this and like that, And it has clamps on either side, so you can have a burette here and a burette there. That's so that you can do a double titration. In other words, you titrate one direction and then you come back. We're not going to do that kind. Actually, that's a little more involved than we need. We're just going to use one side, and it's got a clamp. You just open the clamp by squeezing it, slide it in there, and let it loose, and be sure it fits in its, there's grooves in there. That's what holds it. And then we're gonna, for convenience, now the method might say you're gonna use an Erlenmeyer flask and you're gonna add some of this material, and then you're gonna swirl it. And then you have to put it back down under there and do it again. We're gonna make it quicker. We've got magnetic stirrers, right? There's a magnet in here, a big magnet that spins. And inside there, we're gonna have a Teflon coated magnet that's magnetically coupled to that big thing. And it's going to get it spinning, and that little magnet is inside your container, and it's mixing continuously. So you can let it just sit there. You can add a few drops and watch what happens. Okay. All right. So we're going to do 3 titrations to standardize the base. So the base is going to go in here, and down here for the standardizing the base, sodium hydroxide, we're going to have a standard acetic acid solution. And I don't remember what it is right now. I think the method says it's 0.873. But I think the last time I standardized it, it was a little different. So you'll have the real number when you get to the lab. So you have an absolute amount, a standardized value here. So you measure out a certain volume of this into your container. And if you have the volume of the acid and we know what the molarity is, we can find out the number of moles. And since there's one proton here and one hydroxyl for the base, then we can use this relationship. Like that. Works perfectly well. As long as the number of protons in the acid per molecule is the same as the number of hydroxyls in the base. It's a one-to-one relationship. We can do that. So we know this one, we know this one. We're going to measure out, we're looking for this one, and we're going to determine that one here. So once we do that, we've got those 3 terms and we can calculate the molarity of the base. Now, how do you know when you reach the equivalence point? The equivalence point would be established by a pH meter to be accurate. But if you're going to do a lot of these, you want an indicator, something that changes colors when you get close to the endpoint. And since the titration is going to be of this nature, and I think I drew this one before, pH and the base added. Going to start off here and go like this, and then go like that, and like that. So your pH of the acid would be measured here at around 3.5, something like that. Then you add a little base, and it goes up here. It flattens out. That's the buffer zone. And we'll talk about that. Let's see. Next semester. But the equivalence point is right here in that steep part of the curve. This is when the moles of acid equals the moles of base. For the moles of hydrogens equal the moles, I should put it that way. The moles of hydrogen ions equals the moles of hydroxyl ions. And for a one-to-one relationship, it would be base and acid. But for our complicated one where we have 3 protons in the citric acid, this would be the accurate expression. But that's the equivalence point. Well, notice that the pH is changing really fast. So that's a good thing for us, because if we use an indicator that changes colors up here, then it's just a very short distance as far as volume goes to get from here to here. So the error is very, very small. If we try to do the same thing in here, you know, it'd be a problem. So we pick our indicator is phenolphthalein. I think that's the way you spell it. You used to be able to find phenolphthalein chewing gum in the gag stores, right? And you repackage it and give it to your friend. They chew the chewing gum, they get all that phenolphthalein in the system, and it gives them a bad case of diarrhea, right? But we're not going to use it for that. We're going to use it for color change. So the color change occurs when it's acidic. So we're going to put it down here with our acid, a few drops down there. Doesn't take much. It starts off as clear. When we reach that end point, we get a pink color. So it's a very noticeable difference between clear and pink. It's extremely accurate, actually. Okay, so that's how we tell when we've got enough base delivered in here, when we see that change from acid color clear to just when you add one drop and it changes and it stays changed. You'll notice when we're doing the experiment that as you add base, you get the pink color around the drop that hits it, but once it gets mixed in and the pH stabilizes, then that pink goes away. But as you add more and more and more base, and you get closer to the point, the pink tends to linger longer. So that gives you an indication that you're getting close. The biggest problem, especially for newbies with these titrations, is they overshoot. In other words, instead of adding one drop, they add a mil. And it goes up here all of a sudden. You've way overshot it. And the only thing that particular determination tells you is go to this amount and you better not go fast after that. Okay. So once you've got that volume determined for the base, then you can calculate the molarity of the base. We're going to do that 3 times. We're going to get 3 values for the base and average them. And that's the one we're going to use for subsequent calculations. Because after that, we're going to put a mass of citric acid in here and add some water to it to dissolve it. And then we're going to titrate that again with the same indicator. And do that 3 times. And by doing that, we can determine, since we now know this one, right? And we know that we deliver a volume and a molarity of a molarity base. This now gives us the moles of the base. We divide that by 3, and that gives us the moles of the citric acid. And then we can do our calculation. Okay. All right. So let's do some questions here, work out some problems, get some practical experience with doing these calculations. Okay. All right. Let's see. I'm not sure. Is that part of your the handout?
Homework problem?
This discussion question.
Yes.
Okay, good. We'll do these and then we'll do the pre-lab questions. And I think there's some post-lab questions that you get to do on your own. Okay, so these are the discussion questions. All right. So the first one is calculate the volume of 0.75 molar sodium hydroxide solution required to titrate 25 milliliters of 0.873 molar acetic acid solution. What's the volume? Well, we notice that sodium hydroxide, right, has 1 hydroxyl, and citric acid as one proton. So we can use, and we don't need to do that because the stoichiometry is established here. So now we can say that and this is number one. We can say the molarity of the base times the volume of the base equals the molarity of the acid times the volume of the acid. So what are the values here? Well, the question is, calculate the volume of sodium hydroxide. This is our unknown. So we could go ahead and solve it for that one. Then we can just put in our values. You gotta be sure that you put the values in the right place though. 0.75 molar sodium hydroxide. That's this one. Down here. Molarity of the acid is 0.873. Okay. Notice that the molarities now will cancel. And then the volume of the acid is 25 milliliters. 25 milliliters. Okay. So that's the thinking part. The rest is just number crunching. Okay, what'd you get?
9110.
Mm-hmm. You know, we have three significant figures here, 4 there. So we're going to restrict it to 3. 29.1 milliliters of the sodium hydroxide are required at this molarity to neutralize 25 milliliters of acetic acid at that molarity. That's what we're saying. Now, does that make sense? Well, 25 milliliters of acid at 0.873 is going to produce a certain number of moles, but if the base is less concentrated, it should take more volume. Yeah, so that makes sense. All right, that was number one of our discussion questions. Let's see, number 2. Scroll it up a little bit. There we go. Calculate the volume of 0.75 molar sodium hydroxide solution required to titrate 1.2 grams of citric acid. All right, this is going to be a little more involved. So I'm going to put what we know up here. The mass of the citric acid. Mass of the citric acid equals 1.20 grams. Okay. Molarity of the base that we're using to titrate with it equals 0.75 molar. Okay. We're asked to calculate the volume of that base required. Well, as it turns out, if we know the moles of this citric acid, then we can, by our stoichiometry, we can tell how many moles are required here. Okay. So what's the molar mass of our citric acid? It's not given in the problem, is it? Okay, let's go back up and see if it's given up here. Let's see. Otherwise, we'll have to calculate it. And we can do that. I think we are going to have to do that. Okay, so let's put our formula over here so we be sure and get the right number. C6H5. C6H5O7, correct? I'll go ahead and draw it. This is a tricarboxylic acid. Not mean anything to you, but it's got a carbon backbone here, here, and here. And then each carbon can have 4 bonds to it. So this one, it has a carbon here with a double bonded oxygen and an OH. And this one has a carbon here with a double bonded oxygen and an OH. And this one has another carbon with a double carbonyl, they call that group, and then hydroxyl. Now, we need 4 bonds. This one's got 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. This one only has 2. So it's got a hydrogen here and a hydrogen there. This one's got 3, so it's got 1 hydrogen there, and this one's got 2. So that's your structure. And the protons that we're neutralizing with the base are these right here. That one, and that one. The rest of those hydrogens are virtually locked in place. We can't titrate those. Now let's see if that comes out to the right formula. Let's see, 1, 2, 3, 4, 5, 6 carbons, all right. 8 hydrogens, 1, 2, 3, 4, 5, 6, 7, 8. And 7 oxygens. Let's see, 1, 2, 3, 4, 5, 6, uh-oh. I think there's an OH here. I believe. Let's see if that's right again. 7 oxygens. 1, 2, 3, 4, 5, 6, 7. Yeah, there's a hydroxyl right there. Okay, my mistake. But this one is less acidic than these others, and it doesn't titrate either. Now we could force it, but under these conditions, we're only going to have access to those 3. Okay, now let's do the weight. So we got 8 hydrogens, right? 1.01 for each hydrogen. 6 carbons, 12.01 grams per mole, and 7 oxygens. There we go. So we can calculate the molar mass of that compound. 12.01 times 6. And then 16 times 7. So I get 192.14 grams per mole. Did I do it right?
Yes,
Okay.
that's right.
Okay. So now we can calculate the number of moles of our citric acid. All we need is a little dimensional analysis here. Grams there. Grams need to cancel and leave us with moles. That means the grams have to be on the bottom. So 192.14 goes on the bottom. 1.2, put those to divide. And I get 6.245 times 10 to the minus 3 moles of citric acid. Okay. Now, you get that too? Okay. If we have that many moles of citric acid, how many moles of hydrogen ions do we produce? We produce 3 times that many. That's how many moles of the whole molecule. But we get 3 of these for each molecule, so 3 times that would be 1.874 times 10 to the minus 2 moles of hydrogen ions. Okay? For every 1 hydrogen ion, you need 1 hydroxyl. So that means we're going to need 1.874 times 10 to the minus 2 moles of hydroxyls. Okay? Now, when we say 0.750 molar base, we're saying sodium hydroxide. That's 0.75 molar sodium ions in solution and 0.75 molar hydroxides in solution. Okay. With this relationship where we say the molarity of the base, let's see, equals the number of moles of the base divided by the volume of the base. Okay. So if we're solving for the volume, we do that, we put the moles over here and the molarity there. So we know what the moles are. We just calculated them. 1.874 times 10 to the minus 2 moles. And the molarity is 0.75. Okay. So we should be able to get the volume of the base, and this is going to be in liters. So when we answer the question, we'll give it in both ways. Let me go back down. Come on down here. There we go. There's 2. So, okay, I got this number in here already. I'm going to divide by 0.75. And I get rounded off to 3 significant figures. I get 2.50 times 10 to the minus 2 liters, or 10 to the third times that would be 2.5 times 10 to the first, which would be 25 milliliters. Okay. So what we're saying is 25 milliliters of this base will reach the end point, which is the color change, with 1.2 grams of citric acid. Okay, those were the discussion questions. Now we got pre-lab questions, which is we just kind of flip things around for the pre-lab questions. Let's see. I'm going to write this up here. The accepted, molecular weight of citric acid is 192.14 grams per mole. That's the accepted value. So we're going to have to calculate it again. I'll leave the formula up there in case we need it. This is going to be pre-lab questions. Go. Okay. How are we doing on time? Okay, we're good. So the first one is, we're told to calculate the molarity of a sodium hydroxide solution if 24.3 milliliters were required to titrate 25 milliliters of 0.873 molar acetic acid. Okay, this one, all we need to know firsthand is that sodium hydroxide is monobasic, 1 OH per molecule, and acetic acid is 1 hydrogen per molecule. Then we can use this relationship. Molarity of the acid times the volume of the acid equals the molarity of the base times the volume of the base. Why? Because this tells us the number of moles of acid and this tells us the number of moles of base. And they have to be equal at the equivalence point, which we're approximating with an endpoint. Okay, so let's see. Molarity of the sodium hydroxide. So we're looking for that one right there. Now, what's the molarity of the acid? 0.873. And what volume of acid did we put in our container? 25 milliliters. And by the way, how are we going to do that? We can't use a graduated cylinder. They're not accurate enough. Right? They're plus or minus 5%. That's a mess. We use a volumetric pipette. It's a device that has a long stem here, and then it has a bulb in the middle, and then it has a long stem here with a narrow tip on the end. and it has a mark up here. And that tells you when you fill this up to that mark, you have the volume indicated. In our case, we're saying it's 25 milliliters. And this would be accurate to within a couple of tenths of a milliliter. So how do you use one of those things? Well, there's a procedure. And let's see, I think it was in the other course where I did that procedure. You want to fill it up. There's a procedure for filling it up to the mark. So you use a squeeze bulb up here. It's a flexible bulb, and you just squeeze all the air out of it and then put it on the top and let it go, and it sucks the fluid up into this bulb. And you go up here above that, and then you pull that off and stop it with your thumb. The other type is it has valves in it. Right. It has a valve here, and it has a side valve over here. And you press on this one to suck. It'll say S. It sucks the stuff up, stop, and then you pull it out of your solution and wipe off the end of it, the tip. You don't want anything clinging on the end of it because that's not calibrated inside the container. Then you let it out slowly by pressing this, I forget what the symbol would be on this one. But you press on that button, squeeze it, and the valve opens and lets air in, and it drops. And it takes practice to control that. But you drop it until that meniscus hits that line. Okay? And then it's still out, and you're draining into the container. Then you take another chem wipe. Actually, you don't take a chem wipe. You take this tip and you touch it to the inside up here, the inside of the container, just so that it wicks away any drops hanging on the end. Now you have exactly 25 milliliters in that container. Take it over to your flask and just remove that or squeeze this one right here, and it'll let air back in. And let it drain under gravity. They're calibrated to drain under gravity. This tip is the right size so that it drains slow enough so that anything clinging to the walls has time to drain off the walls. And if you have some left in the tip, that's fine. It's calibrated for that. So all you have to do when you get finished, when it gets down to the end and it stops dripping, you just touch the tip to the inside of your container, wherever you're delivering it, and then pull it out, and you have delivered an accurate 25 milliliters or 10 milliliters or whatever the thing is. Okay, so that's how we got 25 milliliters of this acid in the reaction vessel. Then we added to it 24.3 milliliters of sodium hydroxide. Okay, so now we can calculate the molarity of the base. By the way, is the molarity of the base going to be greater than, equal to, or less than that value?
Greater.
Because you are neutralizing this one with a smaller volume, so it's a higher concentration. Yep, that's right. And let's see, you get the same thing I did?
0.898.
Yep. So that's the molarity of your base as determined by our titration. Let's see, number 2. I think number 2 is going to take a standard base, and we're going to calculate the molar mass of citric acid based upon this titration. Okay, so number 2, we're given 25 milliliters of 0.75 molar sodium hydroxide solution required. Well, the volume of the standard base is 25 milliliters. Now, where did that number come from? Of the standard base. Molarity of the base, 0.75 molar. That number didn't come from one of these. That number came from the burette. We delivered 25 milliliters to reach the endpoint. Okay, for 1.2 grams, the mass of citric acid, 1.20 grams. And we want to calculate the molar mass, so the molecular weight of this citric acid, experimentally determined. So the molecular weight then is going to be the mass of the citric acid divided by the moles of the citric acid. Okay, so we can calculate how many moles of base were delivered, right? That's simple. 0.75 times 25 milliliters. Actually, this is not going to be moles, is it? Because that's milliliters. This is going to be millimoles. And I'll show you how to convert it. The other possibility is just go ahead and change milliliters to liters, right? 0.025, and then it comes out moles. But if you do it this way, 25 times 0.75 is 18.75 millimoles. Well, what's a milli? A milli is 10 to the minus 3, isn't it? So what if I just take that out and say times 10 to the minus 3 mole? There you go. So now we've got the number of moles. If you want to convert it to scientific notation, you move this over, you're taking a value, a 1 decimal point, which is 10, and applying it here. So this should be 1.875 times 10 to the minus 2 moles. Now that's citric acid. All right, how many moles? Excuse me. No, it's not. I'm sorry. That's base. That's hydroxyls. Because we calculated it from the base concentration. So we know that's the hydroxyls. How many hydrogen ions will that neutralize? Well, it'll neutralize the same number of hydrogen ions, right? Because for every one of these, you add one of those and you get a water molecule. But we want the moles of citric acid. Okay. So this many hydrogen ions will represent 3 hydrogen ions per molecule of citric acid. So we actually only have a third of that as citric acid, because a third of that will generate that many hydrogen ions. So we divide this by 3 and we get, let's see, 6.25 times 10 to the minus 3, I believe, moles of citric acid. Let me check myself, 1.875. Yes. 6.25 times 10 to the minus 3 moles of citric acid. Now we got what we need. All right, the mass is 1.20 grams, and the moles of citric acid, 6.25 times 10 to the minus 3 moles equals 192.00 grams per mole. So that's pretty close. Actually, I shouldn't be permitted to use those 2 zeros because there are only 3 significant figures here. So within three significant figures, we're spot on. Which never happens in reality. But you remember how to calculate percent error? When you get your value for citric acid, then you take and do the difference. Let's see. Yeah, it's accepted value minus experimental value divided by accepted times 100. And if you can get a value less than 5% with your first try at titrations, you're doing really good. I've seen them under 1% before. Those, I think it was a bunch of girls. They were real sticklers. Let's see. I think that's all the pre-lab questions. Yep. Okay. So we have two procedures. One, we're going to standardize the base. And the second one, we're going to titrate citric acid. And there are your post-lab questions. We got, let me see, 5 of them. I will say this. Based on the procedure, when you standardize your base, you're going to put a certain amount of base in there that's accurate, but it's such a small amount. In fact, I think it's 10 milliliters. It's such a small amount. It's too small for the stir bar to work really well. So we add some more water. Now, how does that affect the outcome? Doesn't. Because you're basing the titration upon absolute number of moles of acid and base. So you can dilute it if you want. Doesn't matter. Okay. I guess that's it then. So unless there are any questions, let's see. How many did I lose? Or did I get one that I'm missing? Yeah, we got 20 minutes to spare. So floor is open for questions if you have any. How about safety concerns? Should you be concerned with the base or the acid? They're not particularly high concentration, but if you get any on your hand, you got a cut, you're going to find it. So you wear gloves. You wear eye protection because if you splash that stuff in your eyes, it can do some damage. Right? So you need that. And you don't want to get any on your clothes, especially the base. You get the base on your clothes and it might decolor your jean. You have a yellow spot here and there. The acetic acid won't do anything, right? Because we put it on our salads, right? Vinegar. How about what else? Glassware. You can always run into Murphy's Law, right? A dropped beaker will send pieces to the furthest reaches of the universe. And that's my interpretation. You drop a beaker, it's going to shatter. You'll find glass pieces from that beaker in every part of the lab for the next 6 months. So be careful with that. We're not going to use any hot things, right? So that's not a problem. Just skin protection, clothes protection, eye protection. That should do it. Okay. That's all I have to say. Anybody got questions? I'll take that as a no. It's either a no or there are people's names there and there's nobody behind the names. That's possible. Okay. We'll see you next Tuesday and we'll do a review for the upcoming exam and then follow that with a lab, this particular lab. This is a formal lab, so your notebooks have to be ready, have to be prepared. And we'll see you then.